To understand the intricate structure of DNA, it's best to review the basics of the structures of all macromolecules, beginning with this. DNA is just a polymer, which is to say a long string of monomers or smaller molecules that have been covalently bonded together. Mono means one, poly means many.
Hey, Miss Day, do you remember which of the 4 macromolecules DNA is?
It's gotta be protein.
No. Okay, so DNA is a nucleic acid, and that's where it gets its name, deoxyribonucleic acid.
Oh, that makes sense. Okay.
But what I really can't remember is, what are the little monomers called that make it up?
Nucleotides. They're called nucleotides.
Oh, thank you. That sounds a lot like nucleic acids. That will help me remember. Thanks. So we already knew that the monomers of nucleic acids are called nucleotides. What you maybe didn't know, though, is that nucleotides are themselves made up of 3 parts. A phosphate group that's over here to the left, a 5-carbon pentagonal sugar in the middle called deoxyribose, and the code-carrying part of the molecule called the nitrogenous base. They're described as nitrogenous because they have a whole bunch of nitrogen inside of them. And they carry code because there are a couple different nitrogenous bases. For example, this particular nitrogenous base is represented by the letter A, one of the 4 letters of the genetic code. If you string these monomers up, you start making the polymer of DNA, and then you can start calling it by its full name, deoxyribonucleic acid. You can see it gets its name deoxyribo from the sugar that's right in the middle of the molecule. So there's that deoxyribose sugar in this nucleotide, and then in this nucleotide again and again, all throughout the center of the molecule. The deoxyribose sugars are very strongly bonded, in other words, covalently bonded with the phosphate groups, which are then in turn covalently bonded with another deoxyribose, then to another phosphate, another deoxyribose, phosphate, which makes something called the sugar-phosphate backbone of the molecule, connecting sugar, phosphate, sugar, phosphate, sugar, phosphate, running down the back and making the DNA molecule very strong. Then on the inside of the DNA molecule are the actual nitrogenous bases, and here you can see the 4 letters, T for thymine, A for adenine, C for cytosine, and G for guanine, each one with a slightly different molecular structure. Like you can see, this one is sort of big and has 2 rings, and this one's kind of small and has 1 ring. So each of these letters stands for a slightly different molecular structure of nitrogenous base, carrying code T to A to C to G. Now the previous diagram is a little bit misleading because, as Rosalind Franklin and Watson and Crick all found through their research, DNA actually comes in a double helix. So that is a helix with 2 strands running through it rather than just the one. So the diagram we were just looking at looks like half of the molecule right here, but there really is a whole other half which is interacting with that half. So DNA is really a double-stranded helix. Now how did these guys come to find it? In brief, Rosalind Franklin took a bunch of DNA and crystallized it, put it in a crystal, and then used a technique called X-ray crystallography, in which she shot radioactivity at that crystal and then collected on a photography plate how the radiation bounced off. And she's much more sophisticated and smart than I am. I don't really understand how to interpret this picture, but when she looked at it, she said, oh yes, these X's right here represent that there must be grooves in this long molecule, such as the groove right here, a major groove that runs this way, and then a little minor groove between the strands right here. And so she said, all right, this molecule must be in the shape of a double helix. Then it was Watson and Crick who went in and tried to make sure that each of the atoms would fit somewhere in the model, and you can see they were constructing a model right here, a life-size model for themselves of what DNA actually looks like, trying to make sure that all of the atoms had the appropriate number of electrons in their outer shells to make sure that everything was balanced. The end result was our modern-day model of the DNA double helix, as shown right here. When we say double helix, what does that actually mean? Well, if you look really closely at the helix in this diagram, you can see that the nucleotides come in pairs. So here's one nucleotide. This is a cytosine nucleotide. You can see the phosphate group, the deoxyribose sugar, and the nitrogenous base cytosine right inside. And then across from it, though, is a guanine nucleotide. You can't quite see its sugar and its phosphate group, but you can imagine they'd be right in here. Same thing down a little bit lower. Here is an adenine nucleotide, and then right across from it is a thymine nucleotide with its sugar and its phosphate group. So you have one strand going over here, and you have another strand that's exactly opposite it. And it's not random either. As you can see, across from this C right here always seems to come a G. Look, here's another C, and then another G over here. Here's another C, here's another G over here. How could that be? Well, the nitrogenous bases have very particular properties that allow them to interact with each other. First off, adenine and guanine are rather large nitrogenous bases, and so they are called purines. Purines are the large ones. And then cytosine and thymine are a little bit smaller. Look, they're only one ring large, and they are called pyrimidines. It's sometimes easy to mix these up, and so sometimes I draw this little goofy monster to help me remember. So Agua, the purine monster, is the monster that I invented in order to remember that adenine and guanine are both the purines, and so they have these very large double-ringed structures, whereas cytosine and thymine are the small one. Those are the pyrimidines. So Rosalind Franklin knew from her famous photo that the DNA helix must have been pretty smooth. You see in this picture, it's not really that bumpy. So she was able to figure out that the width of the DNA molecule must be consistent. You could never have a purine across from a purine because that would be too wide, nor a pyrimidine across from a pyrimidine because that would be too narrow. But a purine across from a pyrimidine would be just perfect. So inside the DNA helix, a purine nitrogenous base always comes across from a pyrimidine nitrogenous base. Watson and Crick then built on Rosalind Franklin's understanding by trying to figure out, all right, is it the adenine across from the thymine or the cytosine? And here's what they found when they modeled it. They knew that nitrogen, like oxygen, was an electron hog and therefore tended to have a negative charge because the electrons were hanging out there. The electrons therein were getting borrowed from nearby carbons and hydrogens, which were left positive. These partial opposite charges attract and hydrogen bond with each other, and same with the guanine and the cytosine. So inside the double helix, A is always found across from T, and G is always found across from C. And that hydrogen bonding between base pairs, as you can see between the C and the G right here, or the A and the T right here, is what holds the entire double helix together. In summary, there are two different kinds of bonds inside this molecule. There are the strong covalent bonds that run up and down between the sugar and the phosphate in the backbone, and then there are the hydrogen bonds across the center of the molecule, which holds it together.